I'd like to welcome you to a very special night. And no, I don't mean it's the final of Celebrity Traitors, which I think starts at 9 o'clock, but of course to an evening talk from Dr. Louise Lavictoire, the head of science at the Freshwater Biological Association based in Windermere. It's also our last Friends of the Ullswater Way talk of the year. Let me perhaps first of all apologise for those of you who have been hoping to see Louise live in Glenridding Village Hall. The atrocious weather of the last few days, together with roadworks within the area around Glenridding, has made driving at night potentially treacherous. And for Louise herself, who's based near Kendal, it would have been very difficult for her, as well as many of you who may have wanted to watch the talk from elsewhere. So a decision was made a couple of days ago to limit the talk to Zoomers, back to our good old COVID days when all our talks were being transmitted through Zoom rather than holding them in Ullswater's village halls. We hope that the message that the talk would only be by Zoom has got through to everyone. We managed to publish it on our website and social media. So back to the talk. Firstly, as my wife Anne has said, for those who don't want to be part of the record, if you haven't turned off, please do your video screens. And let me welcome Louise. I first met her a couple of years ago at an information meeting in Windermere on the Big Windermere programme, where I was interested to see whether something similar could be set up in Ullswater. I had engaged with her to try to find out whether the Freshwater Biological Association had some basic information about the ecology and fisheries of Ullswater, which of course it does. Last year, she had offered to come and give a talk to us about the role of the FBA, but for various scheduling reasons, it had to be postponed. Today, she has very kindly honoured her commitment to speak to us, and it's very timely because since that time, Friends of the Ullswater Way has created a freshwater ecology project on Ullswater, and 10 days ago we did our first sampling on a shingle beach next to the Ullswater Steamers Pier in Glenridding. 5 other sites around the lake are being monitored, and we hope to expand that significantly over the next few months. Louise's own staff have played a critical role in making this happen, undertaking a first training program with 12 volunteers from the Ullswater Valley, hosted by the Outward Bound Trust in September. Louise herself has worked intensively over the last 20 years on freshwater ecology, notably on saving populations of freshwater pearl mussels from extinction in England. More recently, she has focused on the Lake District Arctic Char Recovery and Management Project, which aims to better understand and restore English populations of the iconic Arctic char. The Arctic char still resides in some of the Lake District lakes, but became extinct in Ullswater about 150 years ago. I understand that in her talk, she will discuss how and why species population numbers have fluctuated over time, and how, by doing research of this sort, she can help us to improve management and conservation decisions today. She'll also present a wider work being carried out by the Freshwater Biological Association and the recent collaboration which I've just mentioned between the FBA and Friends of the Ullswater Way to develop a pioneering citizen science project on Ullswater to monitor water quality. Without more ado, I will pass the floor to Louise for her talk, which with the title Protect the Best, Restore the Rest, Nature Recovery in Fresh Waters. The Zoom is yours, Louise.
Lovely. Thanks very much for that introduction, Tim. I'm just going to share my screen and get this all set up. So I hope you can see the uninterrupted view there of, you're not seeing presenter view or anything, you're just seeing the slide. Is that correct? I also now can't see anyone, so I presume that you can see the correct one.
Yes, I'm sticking my thumb up, but sorry, you can't see
No,
anything.
that's fine.
Yes, you can see.
Fantastic. Well, thank you for that lovely introduction. Yeah, and it's an honour to speak to you all today. I'm going to be focusing on some of the work of the FBA, both past and present. And as Tim said, we've been in contact for a couple of years and exploring ways in which to work together. And I'm really pleased that there are trained lakefly volunteers now within the Ullswater catchment. It's been fantastic working with you so far, and we're looking forward to seeing the data roll in as time goes on. So when I was thinking about what exactly to talk to you about today, I think the original plan last year was to talk about the Ullswater freshwater fish populations, Arctic char populations. And I very quickly realised that actually a lot of the data that we have on the Arctic char populations in Ullswater likely is in our unpublished archive, which the FBA has been going for around 100 years. So our centenary is coming up in 2029. And so actually a lot of the historic data and information we may have had on Arctic char is locked up in that in that archive. And it's not a small archive. We've amassed a lot of data and information over the last 100 years. And so it really would be a full-time job to delve into that and do it justice, really. So I decided to broaden out to the general work of the FBA and how it might be relevant to those in the Ullswater catchment and the wider Lake District as well. So yes, I settled upon the title Protect the Best, Restore the Rest. I was in a meeting recently where we were talking about what we should do to prioritise populations of freshwater pearl mussel. And one of my colleagues from Bug Life came out with this brilliant phrase: stop the rot, protect the best, and restore the rest. And I said that I would absolutely steal that because it's great. So I have. And I'm here to talk to you today about nature recovery in freshwaters and what the FBA is doing. So for those of you that don't or haven't heard of the Freshwater Biological Association, we were formed in 1929 with the remit of understanding and protecting freshwater environments across the world. So in 1884, the Marine Biological Association was set up, based in Plymouth, and their remit obviously was to better understand the marine environment. And so in 1928, several different organisations got together and put forward the idea of a freshwater biological association to carry out very similar work in freshwaters, because there was no one organisation or institution dedicated to the study of freshwaters. So we were set up in 1929, and we have been working ever since, based in the Lake District. And I'm going to go through I'm going to do a very quick potted history of how the FBA kind of came to be, what our research focuses were initially, and then we'll move on to some more contemporary information about what we're doing now. But essentially the FBA we are an advocate for one of the most vulnerable habitats on the planet, and the FBA has long carried out the tradition of doing training courses, passing on our knowledge to the next generation of freshwater scientists, collaborating, ensuring knowledge exchange, and more latterly carrying out species recovery and citizen science programs. So just a quick nod to our heritage. Today I was at the Lakes Forum conference, which is it was the 10th anniversary of the Lakes Forum. That's a group of lake scientists who get together annually to talk about what they've been doing over the past year, and it's a great kind of network to be part of, and I was asked to talk there today about the start of the FBA and how the long-term monitoring programs in Windermere and the wider Lake District started up, really, because the long-term monitoring programs in the Lake District are one of the longest running and Windermere in particular is one of the best known and most studied lakes in the world. So some of this is pilfered from that talk today, but essentially the FBA started long-term monitoring campaigns on many of the Lake District water bodies in the 1930s, not long after the FBA's inception. We focused initially on algal studies, so looking at the base of the food web as it is, and identifying and looking at or researching population fluctuations of freshwater phytoplankton. We started pretty early on as well in about 1935 with some serious freshwater fish research, focusing on species that were within some of the larger lakes. So there was many years of perch, pike, eel, char, brown trout studies carried out, which do continue today, but the actual long-term monitoring of freshwater fish populations actually stopped in 2019. The UK Centre for Ecology and Hydrology took over the long-term monitoring when the FBA and the UK Centre for Ecology and Hydrology, one of their bases is in Lancaster at Lancaster University. We were intrinsically linked for many years, and in about 1989, I think it was, all of the scientists who started out at FBA were transferred onto government contracts with the UK Centre for Ecology and Hydrology, their predecessors. And they carried on the long-term monitoring that the FBA had started in the 1930s, and so they still carry out in many of the lakes in the Lake District the long-term monitoring that goes on in the Lake District. But the FBA also had significant areas of research in bacteriology and also analysing sediment core samples. So these are core samples taken from the middle of some of the Lake District lakes, and it gives a snapshot over time, going back thousands of years, to see what so you can tell from the sediment cores. Things like pollen counts can give you an idea about what tree cover was like in those times. Bacteria and DNA from thousands of years ago can also give an idea about what fish populations and ecological populations were doing historically as well. And some of those core samples still exist today for research purposes. As I was mentioning, we do have a significant archive of unpublished material, so chemical, physical, and biological data. We have over 1,000 boxes of samples, notebooks, pin specimens, bathymetric maps, you name it, we've probably got it in our unpublished archive. And one of the big bits of work that the FBA are going to be focusing on over the next five or so years is unlocking the data and information in our archive, because it really is it has huge potential for research into species recovery, to tell us where the populations were at 100 years ago almost, and where we should be trying to get them back to. So that's going to be a big body of work for the FBA over the next 5 years or so. But I did pick out just a few of the little tidbits of information that I could garner from our annual reports, the FBA's annual reports going back to 1930. So some of the kind of little bits of information that I could pick out were studies on trout within different Lake District lakes, which you can see in the figure here. So this is along the x-axis, you've got age of the fish in years and length of the y-axis. You can see Ullswater there, relatively small trout in comparison to Windermere and Loch Leven in Scotland as well, with Haweswater being even smaller than Ullswater. And one of the sentences in the annual report saying that trout in Ullswater were smaller and more heavily parasitized compared with those in Windermere. There's information about the perch fishery in Ullswater in 1942, and also the study of eels migrating down the Emont yellow eels in 1944. So there is a lot of unpublished data and information out there that could be of interest to the Friends of Ullswater Way and other organisations as well. It's just unlocking it first. And the FBA also has had a long history of training the next generation of freshwater scientists and enthusiasts. Like I mentioned, they started with what were known as the Easter classes in the 1930s, where people from research institutions would come to the FBA for a residential field course of a week and learn from the scientists who were employed at the FBA. And a lot of these, that kind of heritage is continued to today. We have a lot of training courses on different aspects of animal and plant identification. We run courses on habitat modeling. We run courses on river restoration. And it really does span the kind of the generalist and the enthusiast all the way up to professional freshwater scientists as well. So we've continued that legacy as well. So just to give you a very brief overview of what the FBA is kind of structured like now. So as Tim mentioned, I'm the head of science at the Freshwater Biological Association, and I head up the species recovery and the citizen science teams. And then we also have the other kind of major part of the work that we do is learning and advocacy. So as I mentioned, we run the freshwater courses. The last one of this year is actually this week. It's the last day tomorrow. And then our course program will be announced probably in January for the next calendar year. We produce publications as well, publications mainly more recently on identification of particular groups to aid ID of particular groups of both animals and plants, and advocacy as well. So we produce information notes that provide succinct kind of synopses of complex information and turn it into something that's accessible and linked to other resources that people can read around if that's of interest to them. But also run our own newsletter and what other stuff do we do for advocacy? They're the kind of the main. I guess the citizen science side of things is also part of our advocacy as well, and I'll come on to that a little bit later. So I guess the question to really kind of start us off is why freshwaters? So freshwater habitats cover less than 1% of the Earth's surface and yet are home to almost 30% of vertebrate species, including 51% of fish species. So very, very important habitat and something that's critical for most life. Wetlands are vanishing faster than forests, and freshwater vertebrate populations have fallen more than twice as steeply as terrestrial or marine populations. And yet a lot of the research funding and the funding for restoration and conservation tends to go to the better known terrestrial habitats and species. In the recent 2022 WWF Living Planet report, it found that freshwater species populations had declined by 83% since 1970, which is a scary statistic. And really, the 25-year environment plan that we have in the UK, the goals, there's 10 goals within that 25-year environment plan, and the two that are really kind of key to what the work of the FBA is thriving plants and wildlife and clean and plentiful fresh water. So in terms of nature recovery and the title of this talk, which was Protect the Best and Restore the Rest, in terms of species recovery, it kind of depends on a lot of different factors as to how we approach, how we carry out nature recovery. So the FBA isn't an organisation that typically carries out habitat restoration. We usually, we are serial collaborators. We collaborate with a lot of different organisations and each play to our own strengths. We partner with a lot of different rivers trusts, for example. We partner with Natural England, Environment Agency, and other organisations that can affect change. Rivers trusts that we work with and the National Trust carry out a lot of the habitat restoration work that the FBA doesn't get involved in. However, the FBA does have a role in helping, in providing advice, and also in the conservation breeding of different species, some of which I'm going to talk about later on. So in terms of protecting the best, we do run conservation breeding programs, and when I say conservation breeding, that is captive breeding. You might have heard about it. You might have heard about captive breeding rather than conservation breeding. We do carry that out, but we don't really want to be in a position where we have to carry out conservation breeding. That is really the last resort, and really we want to be trying to steer clear of doing conservation breeding because when you get to that stage, really it's gone too far, and that is the last resort of trying to save a species or a population from extinction. So really we want to be trying to be in the position where we are protecting populations rather than trying to restore. So let's take the freshwater pearl mussel as an example, I guess. So where populations are okay but at risk, the idea is to protect. So one example of that is the River Ian in Cumbria, which is the most populous freshwater pearl mussel population in England. It has about 350,000 mussels in that river, and it is it's doing okay. There's juvenile recruitment there, and it's doing okay. So we absolutely need to protect that river. But there are other rivers within England which are not in that favourable condition at all. So, for example, one of the populations that I'm going to talk about a bit later, the River Kent. So the River Kent runs through Kendal, and the population in the River Kent numbers around five individuals. And so really you can't protect that population. Restoration is and conservation breeding really is the only option there. So in terms of protecting the best populations, we would look to measures such as landscape scale restoration or land management, restoring natural processes within catchments, or working with partners to do this, because the FBA doesn't carry out those physical restoration activities. We would look to increase advocacy, education, engagement within the catchment to try and engage people with their freshwater environments and become champions for those environments, and really put in place citizen science monitoring programs so that there is people are regularly engaged with their freshwaters and are the eyes on the ground. Really, they're out there seeing the animals, the species, the habitats monthly, ideally, and it helps kind of with that monitoring side of things to keep an eye on what should be protected. Like I said, in terms of when things start to go a bit too far and protection on its own is really not an option, we need more focus and sustained action. So we would look at what particular actions are needed, because, as I said, conservation breeding on its own is not ideal. It needs to be coupled with good land management, rewilding / restoring the natural processes, as I mentioned in the Protect the Best column, but also looking at what is needed for those particular species. So conservation plans with set targets and actions that are measurable, and look to drive change over short periods of time. And citizen science and professional monitoring can also happen during this restoration side as well. But with some species like the freshwater pearl mussel, it's a protected species, and so you need particular licensing conditions for monitoring freshwater pearl mussel populations, and other species like Arctic char as well. So the citizen science side of things needs to be paired with professional monitoring as well. So in terms of data, then I just wanted to kind of mention the wide plethora of data that is probably already out there and at your fingertips. So obviously thinking about Friends of Ullswater Way and the citizen science program that you are you're running, there is already quite a lot of data and information out there that, and as I'm sure you already know as well, on condition assessment. So where a site is designated, there will be condition assessments which will give you an idea about what the potential impacts on or the particular impacts on the designated site are. You're already carrying out citizen science data, or carrying out citizen science methods, which is giving you access to data that you didn't have before, which is fantastic. And there are a wide number of citizen science projects that you can get involved in, but it's great that you're starting with lake fly and potentially river fly as well. Long-term monitoring programs and continuous monitoring. Some lakes and rivers and water bodies have long-term monitoring programs or continuous monitoring. I don't know of any long-term monitoring programs within the Ullswater Basin other than the Water Framework Directive monitoring that goes on there. But the other thing that's really key as well is oral history, and the FBA historically has had a couple of oral history projects where we've interviewed people who remember habitats as they were when they were younger, and have perhaps fixed point photography information, which gives us an idea about how landscapes have changed over time and how habitats have changed over time. Because the shifting baseline syndrome that we're seeing these days, where people think that certainly people, I guess, my age and younger, think that what we're experiencing now is how it should be, rather than the biodiversity that we had perhaps 80 or 100 years ago. And so that oral history side of things is really important to capture the data that's not written down anywhere, and it's more that anecdotal evidence, but it's really still very important for species distribution and what landscapes used to be like. And I guess, yeah, the question of what does sustainable look like in a catchment, and what are we aiming for? Those are all important questions to ask of the data as well. So just to touch on our citizen science programs before moving on to some of the species recovery stuff that I'm going to talk about. All of the citizen science programs that the FBA runs have the data is open access. So with the lakefly data that you're collecting at the moment, the plan is to make that open access as soon as we've got the database up and online. So Riverfly data, for example, there is an excellent portal where all of the data is at your fingertips for nationally. So all of the data that is on there is freely available to look at and to interrogate, and to, if you've got people who want to do projects for school or university, we're more than happy to share that, and they're able to download that data and information to use for their own research as well. Both of the fly methods, so lakefly and riverfly, provide an early warning system for river invertebrates and the impacts of environmental conditions on macroinvertebrates. So for riverfly, the regular riverfly method, the riverfly monitoring initiative method, is monthly sampling of flowing waters to assess macroinvertebrate presence and abundance. As I said, it's an early warning system, and it's linked to early investigation by the Environment Agency. So very similar to lakefly, a kick sample is performed, and a trigger level is set, which is a numerical value that is worked out when you look at the invertebrates that you get in your sample, quite similar to lakefly, but different to lakefly. The trigger level that is set, if the number that you get is below the trigger level, it's what's known as a trigger level breach. And if that happens, it means that there may be an ongoing incident that needs investigation. So if that does happen, the citizen scientist will do a second kick sample in a nearby area, just to see whether they get another trigger level breach. And if they get a second breach, that is enough to trigger a warning to the Environment Agency. So that can be called in to the Environment Agency, and there are ecology contacts within the EA who will then come out and have a look at what may be going on in the river to see whether they can identify a source of pollution. So this early warning system has been really useful in even over this summer, has been useful in identifying misconnected sewer systems, and where, for example, where people do home improvements and renovations, and potentially the builders don't plumb things back in how they should be, that can lead to sewage and effluent from things like washing machines going straight into rivers, and potentially impacting the biota in rivers. So that can be picked up, and has been picked up by riverfly monitoring. So it's a great early warning system. And very similar to riverfly, lakefly is more seasonal, because monthly monitoring in standing waters, which don't change as quickly as flowing waters. We're still developing the method, but essentially we're asking people to go out seasonally, so 4 times a year, unless you've been told otherwise. Sophie and Emma may have told you to go out more frequently to start with to get some data, so listen to what they say and not me. And then in a similar way to Riverfly, the scores are then uploaded to the what will be the portal online once that's been developed. And that will give us an idea about the taxa within the lake and whether there are any impacts perceived within those particular samples. So this citizen science method is really just getting off the ground. This is the first season of data collection, and it may need some refining over time. But this early data that you guys are collecting is going to be instrumental in showing how the method works and whether the scoring and the method needs any tweaking in future years to make it as useful as possible for monitoring lakes. So another one of our citizen science programs, the Big Windermere Survey, which unfortunately is no longer active. We haven't closed the program down, but we are struggling with funding for the Big Windermere Survey. For those of you that haven't heard of the Big Windermere Survey, this is a large-scale citizen science program which was started in 2002 in response to questions about water quality within Windermere. So Windermere is a mesotrophic lake, which means that it's kind of in the middle. It's not nutrient poor, but it's also not naturally nutrient rich. It's kind of in the middle. But impacts from tourism, from sewage, from land use pressures have meant that it has become more nutrient rich over time. And so there's been lots of action within Windermere over the past couple of decades to improve nutrient concentrations within the lake and to drive those down. One of the biggest ones was introduction of phosphorus stripping at the sewage treatment works, with the main two sewage treatment works within the catchment, which saw vast improvements to the amount of phosphorus that were going into the lake. However, climate change and still pressures of high visitor numbers we weren't seeing a full recovery of the nutrient concentrations in Windermere. And so we wanted to look at collecting data from not just the middle of the lake, which is traditionally where a lot of the data, the water quality data within Windermere were collected, but around the shoreline where people interact with the lake. So the lake is quite popular with wild swimmers, with anglers, with walkers, dog walkers, and these are the places where people interact with the water. And there was really no data on what the chemical and physical conditions of the water around the lake shore, but also within the rivers and the streams which flowed into Windermere and some of the smaller lakes within the same catchment, like Blelham Tarn, Grasmere, Rydalwater. So the survey was set up in 2022 and it consisted of 110 sites from around the catchment. And there is no way with all the will in the world that staff from FBA and Lancaster University, with whom the Big Windermere survey was set up, can get to 110 sites in a day. It's just not possible. And so it was a really great opportunity to engage citizen scientists within the Windermere catchment with their environment. And so we trained people up to carry out the surveys, the water collection, and then the samples are taken to accredited labs for analysis. So the water quality was analysed at Lancaster University, and the bacteriology was done at an accredited lab that was set up for the sheer volume of samples that we were providing for them. So the Big Windermere survey ran for 10 surveys before we had to stop due to funding constraints. And over the summer, we produced a 2-year summary report, which some of you may have seen in the press. It got wide press coverage, both nationally and internationally, and was a great example of how this citizen science program can show real hotspots, and some of them really quite surprising hotspots about where phosphorus, nitrogen, and bacterial concentrations were higher than we would expect in areas that we wouldn't have expected them to be. So there were some hotspots where we kind of realised that there may be issues around very populated areas, or areas that were very popular with tourism and during the tourist season. But there were some areas that were just not on mains drainage, so not on the main sewage system, where septic tanks were the only real possibility of inputs, where we're seeing some hotspots of both bacteria and phosphorus concentration. So this was a really great way to find out what we didn't know, really, and to see whether the phosphorus and the bacterial concentrations changed over time. So this was done seasonally, every 3 months, trying to get in a sample every spring, summer, autumn, winter. And the hope was that it would, over time, become a longer term monitoring program that, as we collected more data points, we would be able to see trends within the data. But unfortunately, it hasn't panned out that way. We had to, like I said, we had to pause it because of funding constraints. But it was a really great example of the power of citizen science and trying to answer a question about where is the water quality a particular issue and what can we do about it. So this is another thing with citizen science programs, I guess, that I would encourage you to think about. What questions are you trying to answer? We knew that we wanted to know about particular water chemistry parameters within Big Windermere Survey, and that's the data that we collected, and it was great to be able to tease apart that and see where these hotspots were, and ultimately to drive action in those areas. So restoring the rest then, species recovery. I'm going to spend the kind of latter part of this presentation talking about some of the specific species recovery work at the FBA. I'm going to talk about the freshwater pearl mussel and our Arctic char programs as well. And it's worth mentioning that before we start talking about conservation breeding, that again, habitat, catchment, landscape scale restoration really is key to this. There is no point in doing captive breeding, conservation breeding, if the source of the issue hasn't been fixed. And that's where the power of partnership really plays out here, because the FBA has the expertise and the experience and the facilities in order to do that conservation breeding. But we could not do half of the work that we do without the power of our partners. So we partner with mainly rivers trusts, like I said, National Trust, and also national parks as well, who carry out the on-the-ground river restoration and catchment restoration that's needed for the freshwater pearl mussel. And we collaborate with them and provide the juvenile mussels ready for release back into the wild, into hopefully restored habitats, to give them a really good chance of survival and the population a chance of recovery. So for those of you that haven't heard of the freshwater pearl mussel before, it's a really fascinating species that's very awkward to breed. I think in terms of awkward species, we started with the most awkward. The freshwater pearl mussel is a very long-lived, very slow-growing freshwater bivalve that is very particular about habitat quality. It needs pristine water quality in order to thrive, and if it doesn't get that, then it starts to go into decline pretty quickly, which is what we're seeing in the vast majority of our rivers in the UK. English and Welsh populations are particularly poor. The populations in Scotland are a lot better, but are also in decline as well. And the species itself is in decline throughout all of Europe. So there are captive breeding programs for the species across Europe also, because they're seeing the similar declines that we are over here. So it's very slow growing, very long lived, can live to be over 130 years old. The oldest freshwater pearl mussel that's been confirmed with, by counting the growth rings on the shell, was 282 years old. So they can be incredibly old, and they can reproduce up until they die. So they don't actually stop producing babies, which is a big benefit, because they really do need every single year that they get if the populations are going to survive. They have an extended juvenile period. So from the diagram here, you can see their very complex life cycle, which involves a parasitic stage on host fish. So if we start, hopefully you can see my cursor actually. If we start over with the adult mussels over here on the left, you can see a release from a female mussel over here, and the larvae of the freshwater pearl mussel is called Glaucidia. So the adults are male and female. They can become hermaphroditic under certain conditions, but generally the adults are either male or female. So in around May time, the male will release sperm into the water column, which is inhaled by females, and that sperm then fertilizes eggs that she's holding within her gills. She broods those eggs for around 6 weeks. It depends upon temperature, but for roughly 6 weeks, and then releases larvae known as glaucidia into the water column. And she can do this in their millions. So their reproductive strategy is one of produce millions, and maybe one or two will actually reach adulthood. So each female can release anything between 4 and 16 million glaucidia into the water column. And these glaucidia, in order to move on to the next life cycle stage, have to come into contact with a salmonid fish. So it's usually in the UK, it's usually a salmon or a trout. In very particular cases, they can also use Arctic char as well, but that's a different story. So they are gill parasites on salmonid fish. So generally populations will use either salmon or trout, and it's usually pretty binary. They will use one or the other, and it depends on the population as to which one they'll actually use. So during the fish's normal breathing cycle, it will take in water through its mouth. It will pass that water over its gills in order to extract the oxygen to breathe. And it's as the fish passes the water, which is laden with glaucidia, over its gills, that the glaucidia, which look like very small castanets, snap shut on the gills of the fish. And as an immune response, the fish will actually encase the glaucidia, the larva, within a cyst, and then the larva will actually grow on the gills of the fish for around 9 months before dropping off into the sediment the following spring, early summer, as a fully formed juvenile mussel. So they do spend a long time on the host fish. Usually in the wild, you would get infection rates of around 10 per fish. In captivity, we can get this much higher, to many thousands per fish, which makes the captive breeding program very efficient. And it has no perceptible effect on the fish at all. The fish continues to grow, continues to be fine, and once the juveniles drop off, the fish is still alive. So it's not within the interests of the freshwater pearl mussel to harm the fish in any way. It's just trying to hitch a ride and grow on that fish and drop off as a juvenile mussel. So for conservation breeding, the Freshwater Pearl Mussel Ark project started in 2007 in response to very fast dwindling populations in England. And the main aim of the project really was to safeguard the genetic stock from each of the English populations. There was around a dozen populations at the time. And also to captive breed, conservation breed from those adult mussels, and to then release those juveniles back out into the wild eventually. As I mentioned, they grow very slowly. So some of these pictures that you can see here, the picture on the far right-hand side is of juveniles that are around about 18 months old. So, and at that time, they're anything between 1 mil and 2.5 mils in length. The picture in the middle is from one of our systems, where those are much older juveniles. So they are anything from kind of fingernail size up to probably an inch in length. And the picture on the far left-hand side is a commercial algal diet that we buy in for the mussels to help them grow fast. So they get fed this once a day. So that's breakfast. So here, hopefully this will work. These are a couple of videos which show, so these are the snapping glaucidia that I mentioned to you. So these have just been released from a female mussel, and you can see they do just look like little castanets, and they're snapping away. And then as they come into contact with a salty environment, which is usually the inside of a fish, they will snap shut and they will stay shut. So you can see I've just added a salt solution to the right-hand side of the image here, and they've all snapped shut and they stay shut. So when glaucidia comes into, let's see what's going on in my algorithm there. whenever they come into contact with that slightly more salty environment that would indicate that they're inside the fish, they would snap shut, and that gives them the best chance of snapping onto the gills and not falling off. And then once they've stayed on the fish for 9 months or so, they will drop off as new juvenile mussels. So this is what they look like when they drop off as newly formed juvenile mussels. So you can see they're quite small. So these down the microscope, again, these down the microscope are around 0.4 of a millimeter in length. So they just look like very small grains of sand. But you can see that their feet, which is the little white tongue-like projection that they're moving around with there, you can see their feet are out and they're moving around, and their feet are covered in these tiny cilia that help direct water flow into their shells, and they feed on very small algae at this stage and grow pretty rapidly. Or rapidly for a freshwater mussel. So by the end of their first summer, they will tend to be somewhere between a millimeter and 1.5 millimeters in length. So that for a mussel is rapid. So just a note on the different systems. So some of these pictures are from our captive breeding program based on the shores of Windermere. So we use different systems depending upon the size of the mussel when we collect them. So the smallest juveniles that drop off of the fish, we put into aquarium systems, which is the picture second on the left there. So these are modified aquaria which have a plastic plate siliconed into the kind of middle of the aquarium, and then into that plastic plate there are 12 to 15 holes drilled into it, within which we put these plastic boxes. So these are mesh bottomed boxes, so they have mesh on the bottom, and we put a little bit of gravel and then the juvenile mussels onto the mesh substrate, onto the mesh, sorry, and then put a second sieve over the top of it, so that the juvenile mussels and the substrate are kind of encased within that box, and they can't get out, they can't go anywhere. And then the water comes into the top of this system. So you can see here the water actually coming into the top of the system, and the only way for the water to exit the system is through the sieve. So the water is forced through the juveniles, providing them with oxygen and food. And then the water comes out of the bottom part of the system here. There's a standpipe, which is just off camera, standpipe in here that brings the water back up and out. So this system can actually be either a flow-through system, which is what this one is at the moment. So the water flows in the top and comes out and goes into the drain, and it only passes through the mussels once. Or it can be a recirculating system that when the water comes out of the aquarium, it goes back down into a sump that is underneath the aquarium, and it's pumped from the sump back up to the top, so it just flows round and round. And that means that we can introduce heat and food into the equation, which helps the juveniles to grow particularly fast. They survive really well in this system because they're provided with plenty of food, warm temperature conditions, so that they grow very fast, and survival is really high. So what we tend to see in Windermere, because we take our water from Windermere, is that as a large lake, it heats up over the summer, and then over the winter months, or over the autumn, sorry, it slowly starts to cool down. So Windermere at the minute is at about 12 degrees C, where we take our water from. And those temperatures are a lot higher than we would see in river conditions where these mussels would usually live. And so the glaucidia on the fish grow a lot faster than they would in the wild. And so what we've seen, certainly over the last 10 or so years, is that the glaucidia are dropping off of the fish after only sometimes a month and a half, 2 months. So they're only spending a very short period of time on the fish, but during that time they've grown fast enough that they're ready to drop off. They're fully formed, and they actually start dropping off in autumn and winter rather than in the spring and summer. So we had a couple of years where we were finding really high mortality rates because the juveniles weren't able to survive over winter. So we needed to come up with a way to keep them alive. And so by putting them in a heated system, which is provided with enough food, they survive really well in that. We can get them over the winter and then introduce them to a normal temperature regime in the following spring and summer, and they get all of the food that they need from Windermere Lake water. So once the mussels have grown enough to move out of this system, so in these aquarium systems, we can get up to 1,000 juveniles per box, per sieve. So if you've got 12 in there, you've got 12,000 juveniles in one aquarium. So they quickly start to outgrow that when they're a couple, anything between 2 and 5 mils in length. And we then need to start moving them out into other systems. So we're tending to try and push them into these upweller systems, which are on the far right-hand side here. And you can see a lot of our systems are really not very high tech at all. So these are just buckets within a large tank, and they're provided with raw lake water from Windermere. And it's a similar kind of concept to the aquariums, where the water flows through a mesh bottomed bucket, and these are flow through systems, so the water flows through once and out, but we can again get thousands and thousands of individuals per bucket. So in any one system that might have 8 buckets there, we can get anything between 10 and 15,000 juvenile mussels in a single tank. And you can see the picture on the second from right is some tagged juvenile mussels they will probably be anything between 10 and 15 millimeters in length, so a centimeter to a centimeter and a half. So we can put them in these mesh bottomed buckets, and they will grow there for a period of time until they're large enough to be tagged, which these ones have been, and then they move into our flume, which is the picture on the left hand side here, which is a mock river. So this is getting them ready to go back out into the wild by introducing them into conditions that they would normally experience in the wild. So they get laminar flow from the side rather than from up or down that they get in the aquarium and the upweller system. They get to bury into substrate, and we vary the water flows over time as well to get them ready to go back out into the wild. So just to kind of summarize where we're at with the program. So we've been running since 2007. We've now, this summer, we released our 5,000th mussel, which was a brilliant milestone to hit. So we've now introduced juvenile mussels into 3 different Cumbrian rivers, and have released over 5,000. We now have over 50,000 juvenile mussels from 7 different populations, most of them English, but one of them Welsh as well, because we're breeding from a couple of different Welsh populations now also. So we've got 7 different populations, or 7 different juvenile populations on site. And we are now getting to the point where we are going to have to scale up our release activities next year, because we've got a lot of juveniles coming through that are of taggable and releasable size. So we're doing well. We certainly made enough mistakes in the first, I think, 10 years to know what we're doing now, and we know what they need. They're very picky as a species, but we do know what they need, and we're now able to sort of upscale the breeding side of things, and need to get them back out into the wild a lot quicker, which is fantastic. Monitoring remains a key part of our strategy as well. So we don't just throw these mussels back out into the wild and hope that they do well. We do tag our mussels as well, so that we can keep an eye on them. So as well as a numbered tag that you can see there, each population has a different colour. The River Ert is yellow. So these mussels are from the River Ert in West Cumbria. So they're yellow, and they'll get an individual number. And then they're also tagged with PIT tags, which PIT stands for Passive Integrated Transponder. And it's a similar kind of tag to what you would put in your cat or your dog. And it's just a passive glass encased tag, which we stick onto the outer shell of mussels with super glue. And then once the super glue is dried, we cover it with a thin layer of dental cement. So dental cement is great for this because it's non-toxic, it's meant to stay moist, and it's very hardy as well. So we cover that with dental cement, and then it means that we can release these mussels back out into the wild. And then the same way that you would scan your cat or your dog if they went missing, you scan the bed of the river and it picks up each individual tag, which has a unique code on it, a unique identifier. And so over time, we can monitor individual mussels and see how many of them are retained on site and whether we lose any to sites downstream as well. So it's a really powerful monitoring tool and has been key in making sure that the areas that we're releasing these juveniles back out into are successful. And it's actually meant that we have stopped releasing into one particular area because we were seeing low retention at that site versus some of the other sites where we were seeing up to perhaps 80 to 90% retention. So that's what we're aiming for. So as well as carrying out the kind of practical conservation work that we're doing, it's also a great opportunity to carry out action-focused science. So some of the different bits of research that we've either done ourselves or have been involved in relate to population genetics. So one of the questions for the Our Forever Kent project, which is the population I was talking about before that has fewer than 10 mussels in the wild, we obviously can't do a captive breeding program with just 10 mussels because the genetic bottleneck would have been far too severe and wouldn't have provided enough adaptive capability of that population to adapt to stresses in future. So we had to make a decision to find a donor population for the River Kent. And so we looked at some of the other English populations that we have to see which ones are genetically most similar, but also genetically diverse enough to have that adaptive ability in future if needed. And based upon the data that we found, we could choose a population that was a suitable donor for the River Kent. We also carried out research to make sure that the mussels that we were using as a donor population would actually use the native fish in the River Kent catchment as well as hosts, because there would be no point in putting mussels back out into the wild if they're not going to use the salmon or the trout as hosts within the river as well. So we carried out quite a bit of research within the Our Forever Kent project to make sure that what we were carrying out in terms of conservation breeding and population augmentation, so putting juveniles back out into the wild was actually the right thing to do. We're also looking into what the effects of stress are on mussels. So mussels do everything very slowly. This particular species does everything very slowly, include dying. So if they are stressed, then they can recover from stress, but can take months to die after a particularly stressful event. So we're looking into what the effects of stress are and whether there are some parameters that are more stressful than others. So for example, with climate change, is temperature a particular stressor that we need to be worrying about? Or is high suspended solids loads within the system, because they're a filter feeder, is that also a big stressor that we need to worry about? And what can we do to mitigate the risks there? We've also looked into different release techniques to make sure that the methods that we're using for release work and that we are getting high survival and growth and good retention at the release sites. And as I said before, monitoring our release success is really key to make sure we're being as successful as we can be. So moving away from freshwater pearl mussels and onto Arctic char. So last year we were successful with getting some funding from the Natural England Species Recovery Programme, and we bid for some money to do some work on Arctic char, and the LD Charm project was born. This is a brilliant project with lots and lots of partners. I think my next slide has all of the partners in it. And again, we're playing to the strengths of each of the partners. So in collaboration with Natural England, we drew up the kind of the four main bits of research that we felt needed to be done to answer questions around what was happening with Arctic char populations, both within the wider Lake District, but particularly focusing on Windermere, because the Windermere population has been the longest studied population, but is also probably one of the most imperiled because of the habitat pressures as well. So there are four main areas that we wanted to focus on, which I'm going to take in turn. So this is the partnership for the LD Charm project. We've got a wide range of institutions that have different levels of expertise, and we also have a couple of industry partners as well. So EchoView and Blueprint Subsea are both industrial partners who are experts in hardware and software of hydroacoustics, so how sound moves through water and how that can help us monitor Arctic char populations. So just a really quick background into Arctic char and where they're found. So this map shows you in the green chart are the ones which are still extant. So these populations still exist. The red char are where they are thought to have now become extinct, and the grey one there in Sprinkling Tarn is a bit of a question mark because eDNA, environmental DNA, picked up Arctic char in Sprinkling Tarn a few years ago, and it was a bit of a surprise because we didn't think that Sprinkling Tarn was suitable for Arctic char. So we're querying that one and doing some more eDNA just to confirm whether that is definitely Arctic char, and so we can do some more assessments on whether that's a natural population or whether they may have been moved there some time ago. So as you can see, the Ullswater Arctic char population, as Tim said, died out about 150 years ago, and we haven't been able to find any tissue specimens for the Ullswater char, unfortunately, which would have told us something about its genetics. So if we take each of those 4 points that were on the slide a couple of slides ago, the first one that we're looking into is Windermere spawning ground use. So it's well known where the Arctic char spawning grounds used to be within Windermere. And so our question here is, are the Arctic char that are in Windermere using the same spawning grounds? So the char in Windermere are split into kind of 5 different races, we think. There's a race which will spawn in the River Brathay, which is at the top of Windermere, the very north of the basin. The Brathay flows into the Rothay, which flows into Windermere. And there used to be periodically a river running population that would spawn within the river itself. And then Windermere is divided into North Basin and South Basin. So you can see the North Basin on the left-hand side here and the South Basin on the right-hand side here. And each of the spawning grounds are split into either autumn spawners, which spawn in the shallows in the autumn. So we're actually starting our research campaign tomorrow to go out and look at environmental DNA samples for the autumn spawners, and there's also some spring spawners, and spring spawners spawn in deeper water up to about 30 meters in depth, and usually in February, March time, so very early spring. So the main methods that we're using for this are eDNA and bioacoustics pilots. So looking at just basically taking the kind of bulk of the work is going to be taking water samples and filtering that water sample through a filter which will trap DNA that is free within the water column. And hopefully we will be picking up lots of char DNA as they come into their spawning grounds to spawn. So we will take eDNA samples over the next 6 weeks at various points throughout the lake, and that will tell us whether it's likely that there are char spawning within some of those historic spawning grounds. We're also looking at taking eDNA samples from other lakes that we are either we suspect they may be in, or where there's been anecdotal evidence to suggest they were in the past. So I was mentioning Sprinkling Tarn before. There's another few lakes that when we've been doing literature searches, have shown that char may be present there as well. So we want to look at those lakes for presence or absence. The second bit of work in Windermere
Louise, can I ask you just to try to bring the presentation to an end, if you wouldn't mind?
Sure, yeah, I will.
Thank you very much.
Last for you, no problem. The second one is spawning ground assessment. So basically, what is the condition of the spawning grounds? This is really to see if we can assess what the main pressures on the spawning grounds are, so that we can hopefully again help mitigate those pressures. The third one is population genetics, so looking at the different populations of Arctic char, both spatially across the Lake District, but also over time, to see how the genetics of the different populations differ, and whether there's been any genetic bottlenecking of any of the populations over time as well, which will give us an idea of what the pressures may be. And developing a conservation plan is the fourth one. It's a couple of little videos here, which I'll just let run. But basically, using all of that data and information. So these are char which are in a riverine environment, male and a female, which went past there. So yeah, developing conservation plans for the species based upon the data and information that we're gathering during this project. And this is an Arctic char attempting to spawn at night, which is when they do spawn, is at night. So we know after this year and after this funding ends that there will be additional work needed, and we are looking to actively fund this. I'm going to sneeze. But in terms of other priority species as well, the FBA is really interested in a lot of different freshwater species, because we know that freshwater species are in decline, and there's some of the sexier species like the beavers and the water voles that you know are pretty well known about, and they're easy to fund because they're cute and everybody gets behind the cute and cuddlies. But I guess the FBA really loves an underdog, and I really love an underdog. So there's all of these other species that are that need our help as well, and which we're interested in bringing back and helping out also. So I'll leave this up there, just ways to get involved. You already are involved, you know, you're doing lake fly and river fly sampling, but there's loads of different ways to improve your knowledge and experience of freshwater habitats and species. And I'm happy to send this slide round in an email, and it, yeah, got some useful ideas as well for other things that you can do to engage with freshwaters as well. There's a great online rewilding course as well, which is completely free through Rewilding Europe. So there's an open university course that's really quite comprehensive, that I would definitely suggest to do as well. I learn loads, and I kind of I kind of think that I know a decent amount, but I learned loads during that rewilding course as well. So I definitely suggest that as a great resource too. We also, FBA has a newsletter which is completely free, so feel free to sign up to that to keep up with the work of the FBA and the progress on both the citizen science and the species recovery side of things. And it also gives you information about publications and courses and stuff that's happening within the FBA as well, including events that we run as well. We've got an annual scientific meeting which is going to be running next June as well. So yeah, if you sign up to that, you'll keep in touch with what the FBA is doing and what our partners are doing as well. Thanks for your time.
Thank you very much indeed. I don't know whether you can make a noise, all of us, maybe not. Shall we try and make a noise? I don't know. Is it possible to stop the screen sharing just so we can see each other's faces just for a short time?
Absolutely. There we are.
Wow, that was fantastic, Louise. Really, you've excited me. We have so much to do in Ullswater. I learned a new phrase as well from you today. I'd never heard of it before. I think you called it, you said the FBA is serial collaborator. And I've heard of serial murderers. I've never heard of a serial collaborator before.
We love a collaboration.
That's a new one too. I love that. Does anyone have any burning questions they would like to pose to her?
There are a couple of questions in the
chat.
I can, unless one from Diana. Diana, would you like to ask it, or would you like me to read it?
Well, maybe I should look at it again. Well, what I was really thinking about is the flooding that's going on right now. And as you were talking about the mussel project in particular, I started wondering, are there some protections that you have against flooding, or this must disrupt some of the work that you're doing? Yes.
In terms of our ability to sample and to get into freshwater environments, yes, it disrupts our work, but mussels and fish and all of the other species have adapted to flooding. Flooding is part of their norm, really. Freshwater pearl mussels need very stable substrates in which to bury. They don't have these thistle threads that a lot of the marine mussels have to cling on to rocks. So through different land management practices and through things like land drainage that brings water into the rivers very fast, and where the river channels have been modified so they're very straight, that can create particular issues with scour. So the mussels can be scoured out a lot easier because the delivery of water is so fast. It scours out the bed, and that's what we're seeing more and more with climate change. So that is a particular pressure. But that happens in those environments that are already quite degraded. So trying to restore natural processes so that rivers behave and respond to floods in the ways that they always used to is pretty key to kind of protecting those environments and making them react in a more natural way, if you see what I mean.
Yeah. Thank you.
Thank you very much. Basak, would you like to pose your question?
About the rewilding, isn't it? Rewild. I'm just going to put a link in the chat. Yeah, I found it already. Yeah, it was a great course. A lot more detailed, and there was exams and everything.
Oh, that's great. Well, thank you very much, Basak. I think she's solved your problem. I'm looking at John Pitchford. I would just like to introduce you to us here, because you're doing a fascinating project which may be of wider interest, I think, to the people listening here. Can you say you are a professor of mathematics and biology and some from York University? Would you say, and with your colleague there, just 1 or 2 minutes about your project, which is so exciting for Ullswater?
Oh, don't put pressure on Early. She's only 1 month into her PhD.
Okay.
Next to me.
Just a minute, very short time, but just...
We're interested in the fisheries of these places. I've got a background in marine modelling and trying to find sustainable ways to harvest marine systems. Mm-hmm. And it often comes down to harvesting smaller sustainable things like pilchards and sardines. And then I found myself on Ullswater and saw that there's a fish memorial in the middle of town and on top of the church. And there was a sustainable fishery, maybe of small things that are interesting, like bendace, which have gone now. For hundreds of years, and we're just curious.
Okay, well, we definitely need to talk, because there was a roaring trade in perchines during the war from Windermere. So perch, huge. So perch, pike, and eels were huge. It was a massive commercial fishery during the war. So yeah, absolutely, we should talk.
So yeah, we're not talking about restoring fisheries, but just wondering how these things worked, just ecologically. With so much rewilding activity going on, it's just fascinating. But I was going to ask about the mussels. Why would I bother jumping onto a fish's gill? Is it so that I can colonize upstream?
Yes, absolutely that. Yeah, because they live in quite fast-flowing rivers. Eventually, the population would just kind of rattle down to the sea. So, parasitizing a fish that moves around, albeit kind of not too far, because they're on juvenile fish, so fry and par, but they will move upstream, and those juveniles will get dropped further upstream. So yeah, absolutely, it's a great way to move around, and also to make sure that the genetics gets moved around as well, so that you're not breeding with your siblings.
Let me take one more question, and then maybe we should close it. Anyone want to pose a question? Then I'd like to say 2 things, and maybe I'll pass the floor to Gordon. I don't know whether, Gordon, you want to say something as our chair. I just wanted to say that thank you very much again, Louise, for programming this and talking to us today. Could you say which charity you would like to people who wish to make a donation for this talk would like to transfer funds to?
Yes, absolutely. No, thank you. That's really kind. Yeah, I chose the People's Trust for Endangered Species because they're a great charity, and they supported some of my PhD work with a small grant when I was a PhD student. So I picked the People's Trust for Endangered Species.
That's great. So we'll circulate that website for information purposes. Thank you very much indeed. We've raised through these talks about £2,000 a year from speakers' choices. So I think thank you for all your generosity over the time. There are two things I wanted to say briefly, and I'll pass quickly the last word to Gordon. One is if anyone who's listening now wishes to be trained as a volunteer for our Lakefly program, please write to me. We're looking to try to expand our operations, and that sort of 12 people have been fully trained who can train others, if you like, because we work in pairs. So if anyone wants to participate in that program, please let me know. And then the second thing is that I'm trying to prepare projects, talks, and so on for next year. If anyone has a speaker or a topic they would like us to include for the next year's program, please also could you let us know. So I'll pass the floor to Gordon, just a few words, and then we'll close.
Yeah, just a few words. Thanks, Tim. So thank you very much, Louise. Just a fascinating talk, and just like to thank you for coming and explaining all of that. And the science is incredible. I'm not a scientist, but I have done research around Ullswater, and going back to 1787, James Clark and his book. In terms of fishing with nets on Ullswater, taking eels away in cartloads when the eels were running. And he mentions the Arctic char as well and their demise, thanks to night fishers on the Goldrill Beck. But what it tells us is that if the Ullswater could support that number of fish and stock that number of fish then I'm sure it could do so again. And that could only be a benefit for everyone. So we look forward to working with you in terms of collaboration. But for tonight, just thank you very, very much. On behalf of everyone, thank you very, very much indeed. And we look forward to further collaborations with yourself and the FBA. I do have a couple of other thanks to make as well, because this is our last talk of the year. So, and a special big thanks to Tim, who's arranged all of the talks. Everyone fascinating in its own right. Everyone, the quality of the speakers that Tim manages to find, how he does it, I have no idea, but we're hoping he'll keep working his magic for 2026 and you'll all come and join us. And also to everyone who's been involved in our project. So two key projects, really, the Wordsworth Way, which we launched in May. Thank you to all of the collaborators on that. And also this Ullswater Ecology project, which Tim is taking a lead on. It's taken a lot of effort, time and effort to make that happen and to get it to the starting line. But the gun's been fired, and we're looking forward to that being a success and to finding more volunteers as well. So Tim's your go-to man on that. And yeah, so that's it. Thank you to everyone else for joining and for listening. It is our last talk, so I wish you well for the festive season. I won't say Merry Christmas, but have a good one, and we look forward to seeing you all on the other side with a fresh round of talks for 2026. So thank you very much, Louise. Thank you, everybody.